So I hope you're clear with the chapter Living World, and after finishing each and every chapter, you please go through the chapter again and again, and you try to solve some of the practice questions also, so that you will be more thorough with the chapter and it will be easy for you to score good marks, because immediately after listening to the class, it will be easy for you to do the more practice questions. After finishing each and every chapter, for that particular chapter, you practice more questions, and that already we are discussing few questions, and you practice some more questions so that it will be easy for you. So hope you're clear with the chapter Living World. And then we saw about the biological classification, that is introduction, how the living organisms are classified. That is 2 kingdom classification, and then 5 kingdom classification. That is we take us 5 kingdom classification, and under the 5 kingdom classification, we saw about in detail about kingdom Monera and kingdom Protista, and the other 3 kingdoms are the fungi, plantae, and animalia. I hope you're clear with the chapters. That is kingdom Monera and kingdom Protista. So today we'll focus on the chapter Kingdom Fungi. So what is the difference between the Kingdom Monera, Protista, and Kingdom Fungi? So we are coming up the category. For example, if you take Kingdom Monera, that is a very primitive thing. And then next comes the Kingdom Protista. And the third kingdom is Kingdom Fungi. So what ways the kingdom fungi, it differs from the other kingdoms that we have seen. So if you take the kingdom Monera, they are all the prokaryotes. So the primitive prokaryotes are all coming under the kingdom Monera. Then comes the Protista. Protista, we saw the all. So what is the main difference that we saw? From the prokaryotes, it progressed to eukaryotes. So there the main difference is all the unicellular eukaryotes are placed under the kingdom Protista. So here still more evolution. So how it has evolved from the, that is we saw in Protista itself eukaryotes. So here this fungi is also a eukaryote. So the main difference between the Protista and the fungi that we are seeing is here the organisms are becoming multicellular. That is, they are progressing from unicellular to multicellular organisms. So most of the multicellular organisms that comes under the kingdom fungi and also plantae and animalia. So here it is a unique kingdom. When you see the fungi, you can see that it is a unique kingdom of heterotrophic. So here they are purely heterotrophic organisms. So there, even in Protista, we saw both phototrophs. Some of them will be phototrophs. That is some plant-like protists we saw, and they are similar to plants. They can photosynthesize. They are autotrophic. But when you come to fungi, all the organisms are heterotrophic. They cannot produce their own food, and they are dependent on other organisms for its food. So they are heterotrophic and they are cosmopolitan. That is, they occur everywhere, and they grow in warm and humid places. So wherever humidity is there, they can grow. Example is bread mold. You could have seen, even if you keep the bread outside for few days, there will be a formation of a mold, fungal growth. There is nothing but the fungal growth. And also orange rot. So you could have seen that is, this is the rot, that is, it is formed on the oranges. So it will grow so fast. Orange rots and mushroom. Mushroom is also a type of fungus. For example, Agaricus bisporus. That is an example of the mushroom that we are consuming. So this is an edible mushroom. And also toadstools. Toadstools are poisonous mushrooms. So these are all the examples for fungi. And also white spots and mustard leaves. See here, these spots, they are all the fungal growth. So there it is due to a parasitic fungus. So the fungus is a widespread occurrence. So they occur in a humid places. So if you keep the food outside for a few days, there will be a fungal growth. That's why we are keeping it under inside the refrigerator. So what is the purpose of keeping it inside the refrigerator? There the temperature is low, so it prevents the growth of the fungus. So the low temperature, it reduces the growth of the fungus. So for preserving the foods, we are keeping it in a refrigerator. So they are basically heterotrophic organisms, cosmopolitan, and they grow in warm and humid places. And the common examples are bread mold, and then orange rots, and the edible mushroom. That is edible fungus is mushroom. And also there are several parasitic fungus which are dependent on the other organisms as a host. And also main storage form in the case of fungus is glycogen and oil. So storage form is glycogen and oil, and they are main decomposers. One of them is they help in the decompose. So they are decomposers. Then the other main things about fungi are they are the sources of antibiotics. For example, you know that penicillin, which is a common antibiotic which is produced from the Penicillium species. Penicillium, Chrysolium, and then there are so many species of this Penicillium that helps in the production of antibiotics. And then some unicellular fungi. We saw that the fungi are multicellular, and the exception is yeast. Yeast is unicellular. So it is a unicellular fungi and is used to make bread and beer. So this, it is a common thing that we are using with the help of yeast, and it is a type of a fungus. And it is, so this you should remember, the unicellular fungi is yeast. The other fungi are all multicellular. Then other fungi, it causes diseases in plants. So it causes diseases in plants and also animals. This wheat rust is a disease which is caused by Phycomycetes. So it is called the rust fungi. It causes the wheat rust. So there are several types of rust which is formed by the Phycomycetes, like black rust, brown rust. So depending upon the type of Phycomycetes which causes the disease. So there are several types like black, brown, and yellow rust by the different species of this Phycomycetes fungus. So they are the sources of antibiotics. And then unicellular fungi yeast is used to make bread and beer, and then other fungi that cause diseases in plants and animals. Example is Phycomycetes, which causes the wheat rust. So these are some of the importance of the kingdom fungi, species of kingdom fungi. So and then we'll see about the structure of fungi. So what do you know about the structure of fungi? Except yeast. So except yeast in the sense yeast differs from the other fungi in many ways. So except yeast, fungi are filamentous. So they are filamentous in structure. And their bodies consist of long, slender, thread-like structures called hyphae. So this thing you have to know clearly. The terms hyphae and mycelium. It is more important here while studying the structure of fungi. So it is composed basically of thread-like structures. It is called the hyphae, and the network of hyphae is known as mycelium. So they branch together to form a structure that is called the mycelium. So for example, here if you say there are two types of hyphae. What do you mean by this? Septate and cenocytic hyphae. So cenocytic hyphae is nothing but they are continuous tubes filled with multinucleated cytoplasm. So what do you mean by this? That is, we saw that what is a hyphae? It is a thread-like structure. So when you, for example, if you see the structure of a hyphae or the mold in the bread, you can see the fine thread-like structures, the initial growth, and then these thread-like structures all they will be connected together to form the mycelium. That is, there will be a more rapid growth. Initially, there will be in only in some places. Then it will rapidly spread to huge areas. So that is the mycelium. The network of hyphae is known as mycelium, and the basic single thread-like structure, we are calling it as hyphae. So if you take the hyphae, septate hyphae means here there is a formation of septum, because there are in between, there is a spectrum, they are connected here, separation here, and in between the connections there are pores. So if there is a septum, you're calling it as septate hypha. So these are the nuclei, cell wall, and pore in between, and this is the septum. But in the case of cenoceptic hypha, how it will be? There is no septate in between, no septum or separation. So how the hyphae is now? So it is a continuous tube. There is no separation, and it is a continuous tube which is filled with multi-nucleated cytoplasm. So there are many nuclei. Here each nuclei is separated by a septum, but here there are no septum, so they are multi-nucleated cytoplasm. They have the many nuclei are present in the same cytoplasm, but here the septum separates the nuclei. So here the cytoplasm will have only one nuclei in each septum, but there it is multi-nucleated. So it is one of the most important questions, that is, what is a cenoceptic hyphae? So is it clear? That is, there are two types of hyphae. One is the septate hyphae, the other one is the cenoceptic hyphae. And another main feature about the fungi is the cell wall is made up of chitin and polysaccharides. So the main component is chitin here. So that is one of the main difference between the fungi and the other kingdom. So the other kingdom, particularly even plants, are surrounded by cell wall, but that cell wall is made up of cellulose. So here the fungal cell wall is made of chitin. So this is one of the main difference. Then we'll see about the other properties. So most of the fungi are saprophytes. So you already know what is a saprophyte. That is, they absorb the matter from dead substrate. So they are dependent on dead and decaying matter for its food. So they are saprophytes. That's why they act as a decomposer. So that they are decomposers. So how they act as a decomposers? They feed on these dead organic matter. So they help in the decomposition of those dead organisms. And some are parasites. Some fungi are parasites because they are dependent on the other living organisms. And some live as symbionts. So what do you mean by symbionts? So if two organisms are dependent on each other, that type of relationship is called the symbiotic relationship. So here the examples are lichens. So lichens is the combination of, that is, fungi and algae. So both are interdependent. So they depend on each other for its survival. So lichens and also mycorrhiza. So mycorrhiza is fungi which are living on the roots of higher plants. So these two are the main examples of the symbiotic relationship of fungi. That is, they live with the algae and also they live in the roots of the higher plants. So these are the main properties of fungi. So next we'll see about the reproduction of fungi. So the reproduction can occur in three ways. One is the vegetative propagation. The second one is by asexual reproduction and also by sexual reproduction. So how these types of reproduction are happening? First one is fragmentation. So you know already what is fragmentation? That is the fungal mycelium. It breaks into many pieces, and that each piece will develop into a new mycelium. Then fission, budding, and also by asexual reproduction, it happens by means of spores. So we'll see in detail about these spores, that is gonorrhea, sporangiospores, and zoospores. And the sexual reproduction mainly by sexual spores. So asexual reproduction also happen by spores, and sexual reproduction by sexual spores. So we'll see about the differences between the asexual reproduction and the sexual reproduction, and what are the differences in the spores. They are oospores, ascospores, and basidiospores. So these are the different types of the sexual spores that is produced by sexual reproduction. So the first one is vegetative propagation. So you know all these steps already we saw in bacteria, and generally about the reproduction, that is by fragmentation. So this is a mycelium. This breaks into smaller pieces, and then these mycelium will form they will grow into a new fungus. And then in the case of budding, the buds will form at one end, and then they will detach from the parent and they'll grow as a new individual. This is particularly happens in yeast, which is an unicellular fungi. And then by fission. Fission also, you know how the fission occurs. First there occurs an increase in the mass, then there occurs a constriction, which divides to form the two cells. So by fission. So all these processes already you are clear with, and we saw in the previous classes. So vegetative propagation is one of the method. And how the vegetative propagation is happening? By fragmentation, fission, and budding. So next we will see in detail about the asexual reproduction and sexual reproduction. So here asexual reproduction we saw it happens by spores such as gonidia, sporangiospores, and zoospores. So what are these? Actually, these spores, that is first we'll see detail about the sporangiospores. So how these sporangiospores are formed? They are formed from sporangium. Sporangiospores are formed from sporangium. They form sporangiospores, and these sporangiospores, they on germination, the spores will germinate and they form an mycelium. This mycelium, then again sporangios. So what is this sporangiospore? So these spores, so this is the mycelial structure, and for example, here they are, this is the sporangium which contain sporangiospores. And this part, this fungal filament bearing the sporangium is called sporangiophore. So this sporangium, it releases the sporangiospores, that is, it releases the spores, and the spores germinate and form a new mycelium. The fungal filament having the sporangium is called the sporangiophore, and this happens endogenously, that is inside. And the sporangiospores, there are two types. One is called the zoospores, and the another one is aplanospores. So what is the difference between zoospores and aplanospores? I hope you are clear with this. Hope it is easy for you to follow, right? So zoospores are called the motile spores, and they have the flagella. So they are motile, and these spores, they are produced in a structure that is called the zoosporangium. So generally the sporangiospores mean sporangium. So they are formed from zoosporangium. Produce zoospores, and aplanospores are non-motile. So sporangiospores which are non-motile are called the aplanospores. So these are the different types of sporangiospores. And then if you take the gonidia, so here also they are formed from gonidia on germination. They form a mycelium. But here the thing is, this mycelium, here they form the structures, that is the gonidiophore. That is called the sporangiophore. Here it is called the gonidiophore. And these, they release gonidia. The spores are called gonidia. And another one main difference is there, the spores are enclosed in a structures that is called the sporangium. So endogenously, they are present endogenously. Here there are no surrounding structures. So there is an exogenous production of spores here. Here it is exogenous production of spores. Here the spores are called gonidia. So I hope you are clear with this, right? The asexual reproduction, it occurs by spores, and here the main spores involved are sporangiospores and gonidia. The sporangiospores are produced in sporangium, and these sporangiospores germinate to form a new mycelium. The fungal filament which has the sporangium is called the sporangiophore. Is called sporangiophore, and sporangiospores are of two types, zoospores or aplanospores. If they are motile and having the flagella, we are calling it as zoospores, and aplanospores are non-motile, and that is because they doesn't have flagella. So what do you mean by that? Where you are saying endogenous, that is they are produced inside this endogenous, then they are enclosed within the structure, that is sporangium. That's why we are calling it as the endogenous. These spores are present inside, but here it is exogenous. So that is the main difference between these two types of spores, that is sporangiospores and gonidia. Okay, is it clear? So next we'll see about the sexual reproduction. So here in the case of sexual reproduction, here there is an involvement of gametes. So there, in the case of asexual reproduction, another main difference is the division mostly happens by mitosis. But here, I hope you are clear with the term mitosis and meiosis. Even we'll see in detail in the next cell division chapter. But here, basically, know that mitosis is a division which results in the production of identical cells. But in the case of meiosis, there occurs a genetic recombination. So, but we'll see in detail about the mitosis and meiosis in that cell chapter. So, in the case of asexual reproduction, only the mitotic division happens, but here there occurs a meiosis. And also the gametes are produced within the structure that is called the gametangia. So here there occurs a fusion of gametes. So depending upon the fusion, we can say that it is whether it is an isogamy, and if based on the different types of gametes that fuse, these are these terms: isogamy, anisogamy, and oogamy. So what is that? So by the structure itself, by seeing this, you can understand that isogamous means they are structurally similar. The two gametes that are here, there occurs in fusion of gametes. The two gametes, if they are structurally and physiologically similar, then they are called the isogamous. In the case of anisogamy, the one gamete will be larger, the other one will be smaller. Usually the male will be smaller and more active. So this is the anisogamy. But in the case of oogamy, what happens here? Here the female one will be, that is, egg will be non-motile, and the male is motile, and that is called the sperm or anthrozoid. So these are the different types of fusion that may happen. That is isogamous, anisogamy, or oogamy. So this is non-motile, and this male gamete fuses with that egg and results in the formation of zygote. So depending upon the type of gamete that fuse together, we can name it as either isogamy, anisogamy, or oogamy. And also based on the different types of fusion, that is, we can differentiate the fungi in different ways. For example, based on the type of spores, for example, oospores, ascospores, or basidiospores. We are classifying the fungi into different groups based on the type of spores that are formed, whether it is an oospore, ascospore, or a basidiospore. And they are produced in the distinct structure called fruiting bodies. So first there is a formation of fruiting spore. In the case of ascospores, it is called the ascocarp. It's called ascocarp. That is the fruiting body. And in the case of basidiospores, we are calling it as basidiocarp. So based on the different types, and also the spores that are produced will be ascospores and basidiospores. So here we'll see in detail about the different process, how the spores are formed. So 3 steps of sexual cycle. There are 3 main steps which are involved in sexual reproduction. The first one is plasmogamy. Second one is karyogamy, and third one is meiosis. So what are these steps? Plasmogamy means fusion of protoplasm of 2 motile or non-motile gametes. So we saw already there occurs a fusion of 2 gametes. The 2 gametes may be either motile, that is, it may be a result in the formation zoospore, or it may be a non-motile. So the gametes may be motile or non-motile. First, there occurs a fusion of protoplasm, then occurs a fusion of 2 nuclei, and it results in the formation of zygote, and which results in the formation of haploid spores. For example, if you take the lower fungi, the gamete will be N, that is haploid, and the gamete from the male nucleus, and this is from the female nucleus, and it will leads to the formation of the diploid zygote, and this leads to the formation of spores. In the case of lower fungi like Phycomycetes and all, here there occurs an immediate plasmogamy and karyogamy. The difference or the time gap between the plasmogamy and karyogamy is less here. Time gap is less. In the case of lower fungi like Phycomycetes. But when you see ASCOMYCETES and BASIDIOMYCETES, so what happens here is, and here they fuse to form only the protoplasm fusion is happening. So the condition will be like this, but there is no fusion of nuclei here, and this leads to, this is plasmogamy, and then comes the karyogamy. So after karyogamy, the fusion of the nuclei will happen and results in the formation of 2N. So this is called synkaryon. The 2N condition is called the synkaryon. Syn means united. So they unite to form the same nuclei. Then this will result in the formation of spores. Haploid spores will be formed by meiosis. So the third step is the meiosis. But this what happens here? There is the long gap between plasmogamy and karyogamy. So as a result of this long gap, there occurs this binucleated stage. So hope it is clear. So here immediate fusion will happen. That is, there is no time gap. So after the fusion of the protoplasm, the fusion of nuclei also happens. So immediately it results in the formation of the diploid state. But here what happens? There is no nuclear fusion. So as a result of it, there occurs a condition that is binucleated condition, and this is called dikaryon. Dikaryon, and the phase is called dikaryotic phase. So this occurs in ASCOMYCETES and BASIDIOMYCETES. So there occurs in plasmogamy, karyogamy, and meiosis. So these are the main steps involved in the sexual reproduction. Clear? So this is what, so in sexual reproduction, two haploid hyphae, they come together and fuse. So here the fusion occurs. Some fungi fusion of two haploid cells immediately result in diploid cells. That's occurring in some lower fungi. But in Ascomycetes and Basidiomycetes, a dikaryotic stage or dikaryophase, that is N plus N or 2 nuclei per cell occurs. This condition is called dikaryon. So is it clear for you now? Later, parental nuclei fuse and the cells become diploid. So this is what is happening in sexual reproduction. Fusion of gametes. So there are different ways in which the fusion of gametes occur. So the first one is the gametangial contact. Gametangial contact. So what is happening in gametangial contact? For example, if there is an mycelium and the same mycelium, different hyphae will be there. That is one fungal hyphae and then different types of fungal hyphae. So one produces an antheridium, that is male sex organ. Antheridium. And here it produces the oogonium, that is the female sex organ. So here the male nucleus with the N, and here it will be a female nucleus with N. So now what happens? The male and female, they fuse together, they undergo plasmogamy and karyogamy. And karyogamy. And to form the zygote, and this is called the oospore. This is a 2N condition, and then it undergoes meiosis to form spores. These are the oospores. That is, this zygote, it by meiosis, it releases spores. They are oospores which germinate to form mycelium. So this is the formation of oospores. What happens is the mycelium, that it happens within the same mycelium. Within the same mycelium, that is different hyphae within the mycelium. One acts as a male sex organ and the other as a female sex organ. So the two nuclei, they fuse together. That is the antheridium. It forms a tube. From the antheridium, it forms a tube and it fuses with the oogonium. Both the gametes, there's a contact between both the gametes and results in the formation of spores, that is called the oospores. But there is another type that is called the gametangial copulation. Gametangial copulation. So here what happens is they are formed between two different mycelia. Here in the same mycelium, you know what is a mycelium? It is a network of hyphae. If it is formed in the same mycelium, but here it happens between the two different mycelium. So here what happens? So this is one mycelium and this is one mycelium. And here these structures are called progametangia. Progametangia, then there occurs a fusion of this gametangia. So here there will be many nuclei. Then these nuclei, they will fuse together to form a zygospore. And again, this zygospore, they undergo meiosis and they form spores. So here the fusion occurs between the gametangia itself. Here the gametes are in contact. Here there is a fusion between the gametangia. And there is another type that is called somatogamy. And in the case of somatogamy, no sex organs are involved. Sex organs are involved. But what happens here? Here only the vegetative or somatic cells are involved. Vegetative or somatic cells are involved in this type of somatogamy. So are you clear with this? Three types of, that is how the two gametes are getting into or fused together in the case of sexual reproduction. So here it happens in the same mycelium, gametangial contact, and results in the formation of oospores. And here the fusion occurs between two different mycelium. It is between the two gametangia. The gametangia are nothing but structures where the gametes are present. So there occurs a fusion between the two gametangia and results in the formation of spores. It is called the zygospores. And in the case of somatogamy, there is no involvement of gametes, but just the vegetative or the somatic cells, they will fuse together and results in the formation of spores. That is the somatogamy. Okay, clear? Then we'll see in detail about the different types of fungi. That is based on the morphology of mycelium. It is about the structure of mycelium and about the mode of spore formation. That is how the spores are formed and the fruiting bodies. The fungi are classified into 4 types: Phycomycetes, ASCOMYCETES, BASIDIOMYCETES, and DEUTEROMYCETES. So these are the different classes. So we'll see in detail about each class of fungi. So the first type is Phycomycetes, which is also called lower fungi. So just you note down the important points that comes under the each fungi category. So that will be easy, or better you put a table. Common things like what about the type of mycelium and what type of asexual reproduction in particular type of the fungi, and what about the type of sexual reproduction. For example, we'll see first for Phycomycetes. So where are they occurring? They occur in aquatic habitats and on decaying wood. In moist and damp places, they occur as obligate parasites on plants. So what do you mean by this? They are mostly aquatic, and where will they be? In the decaying wood, in moist or damp places. Obligate means they are strict parasites, that is they are dependent on plants. So the main difference here is the mycelium is aseptate and sinoseptic. So we saw all the definitions of these terms. So aseptate means there is no septum in between, and sinoseptic, so it is a continuous with multi-nucleated, it is with many nuclei. So example are mucor and rhizopus, that is bread mold that we are commonly saying is an example for rhizopus. And albugo, albugo is a parasitic fungi on mustard that we saw, right? Albugo, and it is also called the white rust, that is it causes the disease white rust. So this is about the Phycomycetes. So for all the fungi, you have to see whether it is aseptate or septate, and whether it is a sinoseptic hyphae or it is a branched hyphae. So that differentiation you have to be clear with it. Here in the case of Phycomycete, it is aseptate and sinoseptic. And also be clear with the example. So many questions may come on this. For example, they'll give us a rhizopus. That is an example of whether it is a Phycomycete or Ascomycete or Basidiomycete and what type of fungi. So we have to be clear with it. So remember the example. You make a tabular column and study so that it will be easy for you to remember things. So here the asexual reproduction is by motile zoospores. We saw what are zoospores and non-motile aplanospores. And they are produced in sporangium. So we saw how they are produced in sporangium. So these are the modes of asexual reproduction in case of Phycomycetes. So if you take the sexual reproduction, fusion of two gametes form zygospores. We saw how the zygospores are formed. That is by the fusion between the two, that is gametangia from different mycelium. So fusion of two gametes, they form zygospores, and gametes are isogamous. So if they are similar, they are called isogamous, and if they are dissimilar, it may be they're anisogamous or oogamous. So we saw all these terms. So hope you're clear with it. So you put a column with what type of fungi it is, and about the asexual reproduction, and about the sexual reproduction, what form of sexual reproduction is happening, and then about the mycelium, whether it is aseptate or septate, and branched or cynocytic, and what are the example. And you write for all four types of fungi so that it will be easy. So this is about Phycomycetes. So common bread mold is the main example of Phycomycete. Then comes the ASCOMYCETE. So what are the main features of ASCOMYCETE? They are rarely unicellular, because we saw the one unicellular fungi, that is yeast. So what is a yeast? It is a Saccharomyces, and it is commonly used in bread, for the manufacturing of bread, and mostly multicellular, and other fungi are all multicellular. Example, penicillium, which comes under the ASCOMYCETE. And mycelium is here branched and septate. So there only Phycomycete, it is aseptate. But all the other, these are all some evolved other fungi. So here they are all aseptate is only in Phycomycete. So remember this in Phycomycete. But other ASCOMYCETE and BASIDIOMYCETE are both branched and septate. So they will have the septum in between the hyphae. So they are saprophytic, that is they are dependent on dead organisms. So they are decomposers, and they may be parasitic or coprophilous. So what do you mean by coprophilous? That is growing on dung. So those organisms which are growing on dung, they are called coprophilous. So the examples are yeast. Penicillium is a main example that comes under ASCOMYCETE. And here the asexual reproduction by gonidia. So there we saw in Phycomycete, asexual reproduction, it occurs by zoospores or aplanospores. We saw the different types of asexual spores, that is sporangiospores and gonidia. But here in the case of ASCOMYCETES, the gonidia produced exogenously. So we saw the sporangiospores are produced endogenously, but gonidia are produced exogenously on a special mycelium called gonidiophores. So I hope you are clear, because we already saw all these terms. So gonidia germinate to produce mycelium. So this is the asexual reproduction in the case of ASCOMYCETES. And then sexual reproduction by ascospores produced endogenously in sac-like asci. So here the fruiting body is called the ascocarp, and the spores are produced in a sac that is called asci. So here, depending upon the different types of spores, the name actually ASCOMYCETE means ascocarp and ascospores. Here the ascospores are produced endogenously. This is the main difference. So examples for the ASCOMYCETES are Aspergillus. So this Aspergillus, it causes many diseases like aspergillosis. That is, it causes an ear pain, And that is, and also for some lung infections, that is, it is like TB, and they produce a toxin that is called some species, they produce toxin that is called aflatoxin, which is carcinogenic, that is cancer causing. Which is some species of Aspergillus. And then Glaviceps, that also causes a disease that is called the main cause of this Glaviceps, it causes ergot disease. Causes ergot disease of Bajra. And Neurospora, know that it is used for the genetic and biochemical studies, and it is called the pink mold, red and pink mold. This Neurospora, and it is mainly used in biochemical and genetic work. And Morella and truffles, they are produced from this Morella is produced from Marcella. And truffles, they are edible. So some fungi are edible and they are used as delicacies. Yeah, both of them. Generally, the study of fungi is called the mycology. So Ascomycota is the name of the class that includes Ascomycetes. Yeah. So Ascomycetes, they are also called sac fungi. So sac fungi examples are Aspergillus, Claviceps. Claviceps causes the disease that is ergot disease of Bajra, and Neurospora is called pink mold, and this Neurospora is used in biochemical and genetic work. And Morella and truffles are edible and they are used as delicacies. Then the next is the Basidiomycetes. So the examples include commonly, mushroom, that is Agaricus bisporus, and bracket fungus. The example for the bracket fungus is called the polyporus. And also puff balls. And they grow in soil, on logs, tree stems, and in living plant bodies as parasites, that is as rusts and smuts. That is called for example, Puccinia, we saw it's called rust fungi. They are called, and smuts is caused by the organism that is called Astilago. So the different, and you have to be clear which organisms comes under the which class. So better you write it in a column, and this we have to be thorough with it, because that is one of the main thing to be remembered in this chapter about the names. So they grow in soil, on logs and tree stems, and in living plant bodies as parasites. So here also the mycelium is branched and septate, same as Ascomycetes. Only it differs in Phycomycetes. Phycomycetes, we saw it as aseptate and cynocetic. In these two, it is branched and septate. So the next is here the main difference here is the asexual spores are generally not found. So first in Phycomycetes, we saw zoospores and aplanospores, and in the case of Ascomycetes, the asexual spores are gonadia. Here there are no, so main difference, you just note it down. The asexual spores are generally not found in Basidiomycetes. But what is a common form of reproduction? Asexual mode is by fragmentation, that is vegetative propagation by fragmentation is happening. And here the sex organs are absent, but plasmogamy occurs by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is a dikaryotic, and it gives rise to basidium. So they form basidiospores, and the fruiting body is called basidiocarp. Here, so what is the process involved here? We saw different types of fusion, that is gametangial contact and gametangial copulation. But here, somatogamy. Do you remember? We saw somatogamy where there is a fusion of vegetative or the somatic cells happens. So here in the case of basidiomycetes, the somatogamy occurs and the resulting structure is eukaryotic and gives rise to basidium. So karyogamy and so these are actually the basic steps in sexual reproduction, that is plasmogamy, karyogamy and meiosis. So karyogamy and meiosis occur in basidium, producing 4 basidiospores, and here exogenous production of basidiospores. So they are not enclosed within any structure. So this is one of the main difference between ascomycetes and basidiomycetes. There the same process. In case of ascomycetes also there occurs plasmogamy, karyogamy and meiosis, which results in the formation of ascospores. There is an endogenous production of ascospores, but here there is an exogenous production of basidiospores, and they are arranged in fruiting bodies that are called basidiocarps. And example is agaricus bisporus mushroom, and this astilago, it causes mud disease, and Phycomycetes is the rust virus, that is rust fungus, and causes different types of rust like black rust, then brown rust, and yellow rust, depending upon the different species of Phycomycetes. It attacks the wheat and brown rust. So these are the main examples for basidiomycetes. And one more feature in the case of basidiomycetes, it forms a septum that is called the doliphore spectrum. Doliphore septum, which is present in basidiomycetes. So what is the difference here? Here in the case of septum, there is a pore in between. And due to the spreading of boundary of the spores, they form a dome-shaped structure. That is spreading of these boundaries, they form a dome-like structures, and they form a septum that is called the doliphore septum in the case of basidiomycetes. And also one more thing is there is a formation of clamp connection here. So what is happening? It occurs between the vegetative or somatic cell. For example, here are the 2 vegetative cells. So here what happens? There occurs an fusion. And then plasmogamy. Then there occurs a clamp-like structure, that is between here to here. There will be a connection, tube-like connections will be there. So there is a transfer of material. So mononucleate will become dinucleate. So through this clamp connection, there occurs a transfer of nuclei and they become a binucleated structure. Then there occurs a formation of the basidium. Here. So 2 nucleated structure, it forms a basidium. So this is basidium, and then this nucleus. Here it moves to these structures. Here the nucleus to the structure, and then they undergo meiosis. It releases spores. So it results in the formation of basidiospores. So there, here the clamp connection is one of the main feature in basidiomycetes. There occurs in formation of doliphore septum here. So these are the main features of basidiomycetes. Then the next type is deuteromycete. So it is also known as imperfect fungi. So why it is called imperfect fungi? Because only their asexual or the vegetative phases are known. So, but there is no sexual phase for this type of fungi. So when perfect sexual stages were discovered for a particular fungi, first they will put under the category of imperfect fungi or deuteromycete. So when the perfect sexual stages were discovered, they were often moved to ASCOMYCETES or the BASIDIOMYCETES. So when the characteristics are known, they will go into the further higher category. That more closely related category, they will be moved. So it is also possible that the asexual and vegetative stage have been given one name, placing under the deuteromycetes, and the sexual stage another name, placing under another class. So when the linkages were established, the fungi were correctly identified and moved out of deuteromycete. So only imperfect fungi will be under the category of deuteromycete. Is it clear? So the deuteromycete reproduce only by asexual spores. That is gonidia, and mycelium here is also septate and branch. So these are the main differentiating points. And some are saprophytes or parasites. Majority are decomposers. So the deuteromycetes are decomposers of litter, and they help in mineral cycling. So these mainly act as decomposers. So examples are Alternaria, Colletotrichum, and Trichoderma. For example, the athletic foot disease in human, and also plague disease of potato, that are all caused by these species of deuteromycetes. So these are the different types of fungi. It is Phycomycetes, ASCOMYCETES, BASIDIOMYCETES, and DEUTEROMYCETES. So hope you are clear with it. So we'll see a few questions related to this. So mode of nutrition in fungi is whether it is parasitic, saprophytic, autotrophic, or heterotrophic. So which one will be the correct answer? Yeah, option D. Good. Good, Nandini. So it's easy for you. So I am sure that you know all these terms, so no need to explain. So the answer is answer D. That is heterotrophic. So all of the following are fungi except yeast, penicillium, plasmodium, and Phycomycetes. So which is the correct option? Yeah, good. Option C. Because yeast, sonau, penicillium, and plasmodium is a protozoan which comes under the protista. All the others are fungi. Yeah, good. Option C. Cell walls of all fungi consists of the polysaccharide. I think it's an easy question for you. Yes, option A. Chitin. Chitin is the main characteristic feature of fungi. Fungal cell wall. Cynocytic hyphae is uninucleate hypha, multicellular hypha, multinucleate hypha without septate, and hypha in xylem. So fourth correct answer is option C. Yeah, good. Multinucleate hypha. So which of the following is the correct sequence of the three steps involved in the sexual cycle of fungi? Whether it is mitasis, meiosis, and fertilization, plasmogamy, karyogamy, and meiosis, meiosis, plasmogamy, and karyogamy, karyogamy, plasmogamy, and meiosis. So which is the correct option? Correct order. Timon. Yeah, it's B. Correct, Timon. Good. Fungi are classified on the basis of morphology of mycelium, mode of spore formation, development of fruiting bodies, and all. Yes, Nandini, good. All. By all these modes only we are classifying. So that is the correct option. So it's easy for you, no need to explain all this because we saw in detail about all these. Members of Phycomycetes are found in aquatic habitat, on decaying wood, on moist and damp places. They are found as obligate parasites on plants. So which is the correct option for this? Yes, option D is correct. Because they are found in all the habitats. That is in aquatic habitat, decaying wood, moist and damp places, as obligate parasites on plants. So all of the above is the correct option. Not these things. So the next question is, which of the following is used extensively in biochemical and genetical work? Whether it is agaricus, alternaria, neurospora, and mucor. So which is the correct option? Yeah, good. And then the option C is the correct option. That is the neurospora. Here agaricus, you know that mushrooms, agaricus bisporus is an edible mushroom, and alternaria is an example for deuteromycetes, and it causes the disease that is called the early blight of potato, and mucor and rhizopus so under the bread mold. But neurospora is extensively used in biochemical and genetical work. Okay, the next question is, which of the following is false about deuteromycetes? They reproduce only by asexual spores, that is gonadia. Mycelium is branched and septate. They have only parasitic forms. They have no sexual stage or a perfect stage. So which of the following is false? So which is false in this? The case of deuteromycetes. So here they reproduce only by asexual spores is a correct. Yes, option C is the correct option. I hope you are able to follow. And mycelium is branched and septate, and here they have no sexual stage or the perfect stage, because if it is perfect, they are classified into some other categories. So they have only parasitic form. This is the false about deuteromycetes. So is it clear then? So any doubts regarding this? So I hope you are clear with the kingdom fungi. So what are the things that we saw about the fungi? What are the characteristic features of fungi, and about the mode of reproduction, that is vegetative propagation, and asexual reproduction and sexual reproduction. So vegetative propagation, we saw about fragmentation, budding, and also fission, and under asexual reproduction by the formation of spores, and then under sexual reproduction also different types of spores, oospores, ascospores, basidiospores, and then about different types of fungi, that is Phycomycetes, ASCOMYCETES, BASIDIOMYCETES, and DEUTEROMYCETES. Okay, so is it clear then for everybody?